Hey guys, this is my video assignment for Organic Chemistry Lab 6BL. I'm Amy Becklin, and today I'm going to be showing how you can use purple cabbage as a pH indicator. So the supplies that we're going to be using today to perform our experiment include glass containers. In this case, we're going to be using wine glasses. Those are going to hold our different acids and bases, and they're also going to hold the indicator solution that we make from our purple cabbage. So the purple cabbage is going to be the source of the pH indicator. And then we have baking soda, which is basic, and we know that baking soda has sodium bicarbonate, which has a pH of approximately 9. And our second base that we're going to be using is Windex, which contains ammonia, which has a pH of approximately 11. So it's slightly more basic than our baking soda. And then our first acid that we're going to be using is apple cider vinegar, which has a 5% acidity and a pH of approximately 5. And then we're going to be using our lime to have citric acid, which has a pH of approximately 3. So this is going to be more acidic than our apple cider vinegar. We're also going to be using tap water today as our solvent for our indicator solution. And this tap water might be a little basic, so it won't be perfectly neutral when we make our solution because the water in IV is gross. So pH is a logarithmic measurement of how many hydrogen ions are in solution. So with a low pH, we're going to have a lot of hydrogen ions in solution, and that's going to correspond to an acidic solution. And then for a basic solution, we're going to have a high pH, and that's going to correspond to a low number of hydrogen ions, which will be something like our baking soda. So we're going to be tearing the cabbage into small pieces, and we're going to get approximately 2 cups of cabbage tears. And the purpose of tearing the cabbage is so that we can increase the surface area and ultimately have a more effective reaction in which the pigments in the cabbage are broken down by the hot water. So now we're going to be adding our cabbage pieces into boiling water, and then we're going to let it boil for about 10 to 15 minutes. And what that's going to do is that's going to break down the cellulose in the plant and release the anthocyanins, which is the pigment that's present in red cabbage. While this is boiling, we're going to go prepare our acidic and basic solutions. So first we're going to start with a little bit of water to dilute our solutions. So here we have our 4 different solutions with each different pHs. Okay, so now we're going to remove the cabbage solution from heat, and the reaction is going to continue to occur because it's still hot, but we're going to let it cool for 15 to 20 minutes so that we don't burn ourselves. So as you can see, the cabbage has slightly changed colors because of the loss of pigment into the water. So in the first solution, we see that with the lime and the cabbage solution, the color has changed to a dark red pink, and that's because it's the most acidic. In the second solution of acid with the apple cider vinegar, we can see that it's slightly pink-orange because of the orange nature of the vinegar we used. However, it's clear that it's less acidic than the lime. With the sodium bicarbonate solution, we can see that the cabbage has turned it slightly blue, which makes sense because it is the lesser of the bases. In the final solution containing the Windex and the cabbage solution, we can see that it's turned a dark green, dark blue color because it is the most basic solution. The pigment found in purple cabbage is called anthocyanin. The basic form of anthocyanin has resonance forms, and they are highly conjugated. We know that the presence of conjugation in a molecule will make it incredibly stable, meaning that it's low in energy. Energy and wavelength of light absorbed are inversely proportional due to Planck's equation, so its low energy corresponds to high absorbed wavelengths of red, which will appear blue-green in color because the color opposite on the color wheel of light will be observed. The neutral form has no resonance nor charges, which means that it's pretty average in energy and stability. The solution appears purple and absorbs yellow wavelength for the same reason as described for the basic form. The acidic form has a positive charge on the oxygen molecule and does not participate in resonance, so it is not stable and fairly high in energy, which will be reddish pink. absorbing greenish wavelength, which corresponds to a lower wavelength than the neutral and the basic for the same reasons as I described above. It's important to note that the cabbage solution appears blue rather than purple because of the use of tap water, which is slightly more basic than distilled water. If one wanted to fix this error, they could use distilled water instead of tap water to have perfect results that correspond to the color expected. Cheers.